Hi friends, you're back with me, Professor Girish Kukreja. Today we are here to know about the consequences of contamination, right? The contamination which takes place most of the times when you are in the laboratory, when you are preparing a pure culture. That is one kind of contamination that is going to create a problem for your one plate or one flask. But try to understand and imagine a particular scenario where you have a fermenter which is having thousands of liters of what you call as your fermentation medium, and that is now getting contaminated. It can seriously result in lot of great losses. So talking about the contamination, like what exactly this contamination will do. Many of the times, like we are like, ho hi kya jayega? Like what will happen, right? So let us try to what you call as summarize and see that what could be the possible consequences of contamination of a typical fermentation. Okay, so the first one, it says that it has to now support the growth of both the organism. So now you have an organism which is producing your product, and you have an organism which is your contaminant. So your nutrients are now getting diverted to both. So your producing organism is getting less nutrients, so less growth, right? So it has to support the growth of the both. This doesn't seem to be very serious, but then we have more serious consequences. A continuous fermentation, like many industrialists are involved in a process of continuous fermentation. Remember, we had seen the chemostat or the turbidostat. In this, your contaminant organism may outgrow the producing strain. So now, ultimately, after a certain period of time, you will have your entire fermentation vessel filled only with the contaminant. Now you don't have your producing organism, so no product at all, a serious loss. So we have a contaminate which will be contaminating your final product, like talking about SCP, the single cell protein. Now what you do there, you have a fermentation broth, you have your organism where the organism itself is your product. So single cell protein mein you have organism which itself is the product. So when you are filtering it or when you are centrifuging it and separating your cells, you are surprised to see that there is no of your product, or it is totally contaminated with all other types of organisms. So your entire product is lost probably. Now we have producing the compound which may make the extraction difficult. So the contaminant which is sitting in the fermentation broth is not going to sit just like that. it will do something. You don't just sit like that. So contaminant also, when it is in the fermentation broth, it is doing something, and probably it is producing certain compounds which are going to make your extraction process difficult, which is already much complicated. We had seen that downstream processing of the fermentation product is really a complex matter, and this makes it more complex. It will degrade your desired product. Come on, you are producing an antibiotic, and imagine that there is no antibiotic at the end of the fermentation. Obviously, the contaminant will have to be such that it can overcome the inhibitory effect of that particular antibiotic. Obviously, we have seen that many of these organisms can degrade that antibiotic. The most common example is that of beta-lactamase, right? Beta-lactamase producing organism, if it enters your beta-lactam fermentation, it will degrade that beta-lactamase. So now you have no active antibiotic present there. So be careful. And then you have this small little one, which actually this little one has moved the world at large. But then we have these bacteriophages. If this now finds place in your fermentation, like if it is a bacterial fermentation, if you have a bacteriophage coming in, you know that your producing organism is dead. So it will cause the lysis of the cell, and therefore you will find that there will be no production of your product. So to summarize, it will have to support the growth of the both. In a continuous fermentation, it will completely outgrow and displace the producing organism. It will contaminate your final product, like that in your SCP. It will produce compounds which will make the extraction difficult. It will degrade the desired product, like that in beta-lactamase, and a bacteriophage will lyse the producing organism. So we need to prevent this, right? So we have talked about the problems. Let's soon talk about the solution. So stay tuned with me, Professor Girish Kukreja. Thank you so much.